So, good morning, good afternoon, and good evening. It is with great pleasure that I welcome you all here today for this webinar on optoacoustics and optical complementary modalities for molecular imaging. I'm Dr. Tim Devling, Director of Sales and Applications at Ithera Medical, and I will be moderating this session. The webinar will last for about 45 minutes, with a break. In tradition, we're going to be having 3 speakers and time for a panel discussion at the end. So we really encourage questions. If you maximize the Go-To tool, you can see a question box where you can type them in and we'll address them towards the end. This webinar will also be recorded and we'll send you a link in the next few days. Now, as there are a number of new faces in the audience, I'll begin by giving a short introduction to the MSOT optoacoustic technology and a general overview of the types of imaging being performed before handing over to the speakers. The system used in the following research is the MSOT Envision, with MSOT standing for Multispectral Optoacoustic Tomography. To demonstrate how this works, picture the mouse here in the imaging system. It's surrounded by a 270-degree arc of up to 512 ultrasound detectors with large surface area and high sensitivity. Pulsed laser light is being delivered around this arc to illuminate the animal in 360 degrees. Photons from this pulse are scattered and absorbed as they transmit through the tissue, and when absorbed, this causes the generation of an ultrasound pulse, which is detected and reconstructed into an image by the system. Now, by altering the wavelength of light used, it's possible to identify specific absorbers in the animal and visualize the distribution at high resolution in deep tissue of those absorbers. For example, here we're looking at hemoglobin, oxyhemoglobin, and IR dye. So the unique MSOT capability is its ability to visualize and quantify chromophore distribution, as mentioned, at high spatiotemporal resolution in deep tissue. So I suppose the next question is, what exactly absorbs that light? So, as with optical, to ensure good light penetration, the system uses the near-infrared window. Here, the laser ranges from 660 to 1300 nanometers in 1 nanometer step sizes. This allows detection of endogenous absorbers. As you can see here, the spectra for those absorbers within the system, which include HB, HbO2, melanin, and in the further near-infrared window, lipids and collagen. It also allows the detection of exogenous contrast agents, which we will focus of this webinar. And the general rule is that if it absorbs in this region, it will give signal. Agents used include the organic dyes, such as the cyanine dyes or other fluorophores, bioinorganic particles, such as gold and carbon-based nanoparticles, and chromophores, which can be expressed within cells, for example, like IRFP 720. So how can this be applied in imaging? Well, if you consider the endogenous signals, so for example, HB and HbO2, as well as getting pretty good vascular images for anatomical imaging within the animal, it also gives you the capacity to look at tissue oxygenation. Melanin obviously allows you to detect melanoma, for example, transiting through lymph nodes within an animal. The ability to detect lipids could be useful for imaging lipid dysfunction, so for example, lipedema, while detection of collagen is useful for the detection of fibrosis or muscle injury. But when it comes to exogenous agents, there are a number of uses. From simple non-targeted dyes to look at organ clearance, so for example here, looking at clearance through the kidney structures, to expression of fluorescent proteins within cells to look at the distribution of cancer, so for example here is a glioblastoma in the brain of an animal. And with contrast agents, it's also possible to label peptides to target specific markers expressed in cancer, for example here, orthotopic pancreatic cancer, or to look at enzymic process involved in example, the tumor microenvironment as shown here, so activatable agents. So with that summary, I want to introduce our speakers today. Our first speaker is Dr. Judith Weber. She completed her PhD in the Vision Lab at the CRUK Cambridge Institute, where she worked on activatable contrast agents for the detection of oxidative stress in cancer. Our second speaker is Alexa Hasenbach, who's just completing her PhD at the University of Münster, where she will speak about her work looking at optoacoustic tomography of inflammatory activity in early stages of arthritis using a specific tracer. And our last speaker is Dr. Cheryl Roberts, who's a research scholar at Memorial Sloan Kettering Cancer Center in New York, and she's going to talk about her work imaging the glucagon-like peptide 1 receptor with optoacoustics. So what I'll do now is I'll hand over to Judith, and she can continue this discussion.
Can you see my screen?
1 second, Judith.
Sorry.
There we go.
Okay, thanks. Hi, hello everyone, also from my side, and thank you very much, Tim, for the nice introduction. So I will continue now with an example of an activatable contrast agent for the detection of oxidative stress in cancer. So why do we want to image oxidative stress in cancer? Well, as you are all aware, oxygen is crucial for our lives, as our cells need it for the energy production. However, the use of molecular oxygen also bears some risks, as by accepting extra electrons, oxygen can get converted into highly reactive oxygen species, short ROS. Among those are the superoxide anion and hydrogen peroxide. As the name already implicates, these oxygen species have a really high reactivity, and thus they can cause damage to biomolecules in our body, for example, the DNA, proteins, or lipids. Surprisingly, low levels of these ROS are actually essential for our body, as they are involved in cell signaling and maintaining cell homeostasis. However, our body ensures under normal conditions that these levels of ROS are kept low to prevent cellular damage, and this is done by so-called antioxidants, which are able to scavenge the ROS. Unfortunately, under certain circumstances, the ROS levels increase to a pathological concentration, at which they play an important role in the initiation and progression of cancer and other diseases, and they can even lead to cell death. So looking more deeply to this two later parts of this concentration gradient, you can see that the cancer cells enabled to grow and develop must tightly balance the ROS levels so that it supports their growth but doesn't lead to cell death. To sum up, we can say that there exists a really complex interplay between redox biology and cancer development, and that ROS are involved in every stage of the development process. But a whole story about what is causative or simply correlation is still far from being understood. And thus, we hypothesized that if we could image the redox stage throughout the cancer development process, we could not only shed light on the complex redox biology in cancer, but also find a new tool for early cancer detection, prognosis, and treatment response. However, there are some challenges associated with imaging reactive oxygen species, as the reactive oxygen species are highly reactive and thus have a short half-life due to their fast conversion, which also results in a very high heterogeneous spatial and temporal distribution. Additional to this, there's a lack of imaging tools, since most of the ones used at the moment do either disturb cellular integrity, generate ROS themselves, lack temporal and spatial resolution, are just not applicable for in vivo imaging with a sufficient depth penetration. And thus there are two areas which we wanted to address, and one was to use an in vivo imaging modality with sufficient depth penetration and high temporal and spatial resolution, which we decided to use photoacoustic. And then the other one was to find a way how we can detect one specific reactive oxygen species associated to cancer. And hereby we wanted to focus on a contrast agent detecting hydrogen peroxide, as hydrogen peroxide is one of the most abundant reactive oxygen species with a rather long half-life. So how did we approach the design of this contrast agent? So in general, what you want from a contrast agent is that it has a rather good bioavailability and good pharmacokinetics, and that it has a low accumulation in the non-targeted tissue, but a high accumulation in the targeted tissue, which means in our case, the cancer tissue. And as soon as it accumulates in the cancer tissue, what we want is that it reacts with the pathological concentration of hydrogen peroxide and allows us a readout of this. So from a chemical perspective, this looks like this. So we decided on using the cyanide backbone, which you can see here in yellow, because this backbone is known that it absorbs in the near-infrared range and has a signal profile which is distinguishable from the intrinsic signals of our body. Furthermore, we wanted to obtain an increased accumulation in the cancer, so thus we added a targeting structure. And as a preliminary cancer targeting structure, we added deoxyglucose, as cancer cells show in general a high proliferation rate and thus a higher glucose uptake, which should go in line with a higher uptake of our contrast agent. Furthermore, we also needed a part that reacts with hydrogen peroxide, because this is the reactive oxygen species which we wanted to detect. So therefore, we used this baronic ester functionality, and the reaction of this group with hydrogen peroxide triggers the cleavage of this group, which then leaves us with this uncapped dye version. So as you can see here, I wrote linker, and this linker has actually an important role because it needs to trigger a signal change as the dye backbone stays the same. So we investigated different linker structures which connected this reactive group with the dye backbone and found that the piperacine group actually does a great work in inducing a signal change if this group gets cleaved. So meaning that our final structures look like this. In the following, I will always highlight our starting contrast agent, the capped dye, which has the hydrogen peroxide group still on, in yellow, and the uncapped dye, which results after reacting with hydrogen peroxide, in purple. So having synthesized these two structures, we first investigated their optical profiles. And as I hope you can appreciate from the graphs A, B, and C, that the optical profiles of the two versions of the dye, so the capped and the uncapped dye, can be clearly distinguished with the three imaging modalities: absorption, fluorescence, and photoacoustics. And also the photostability looked rather promising for both dye versions. So having found this, we then investigated if we can actually monitor the reaction of the capped dye with hydrogen peroxide into the uncapped dye, and by using pathological concentrations of hydrogen peroxide. And as you can see here, we always started with the yellow absorption profile corresponding to the capped version of the dye and incubated it with hydrogen peroxide, and you could see that the spectrum converts into the purple spectrum. And this was visible with all the three imaging modalities, again with absorption, photoacoustics, and fluorescence imaging. And looking at the reaction kinetics, we could also see that this reaction happens rather fast in between the first 10 minutes. So we further then wanted to see if the reaction is selective for hydrogen peroxide, meaning that this reaction is not caused by any other reactive oxygen species. So we incubated the dye as well with other reactive oxygen species. But as you can see here, the starting peak here only gets converted into the product peak with hydrogen peroxide and not with any other reactive oxygen species we have tested. So to summarize, we can say that we have synthesized a contrast agent that is specific to the reaction with hydrogen peroxide and can be monitored with 3 different imaging modalities. So knowing this, we then continued and checked the serum stability, the protein binding, and cellular toxicity with different cell lines. And for all the tested cell lines, we did not see any toxic effects. Furthermore, we studied the cellular uptake and cellular localization of the dye in 2 human breast cancer cell lines, the MDA-MB231 and the MCF7 cell line. And as shown here in the graphs, you can see that the uptake happens mostly in the first 15 to 30 minutes, and that the dye actually accumulates in the cytosol of our cells. So having investigated this in vitro profile, we then went ahead and did some in vivo studies in MDA tumor-bearing mice. And as shown here in the iris, as well as in the MSOT images, you can see that the dye accumulates rather fast after the injection in the tumor, as well as in the liver, kidney, and spleen. But it specifically accumulates only in the tumor and in the liver, as it persists there for over 24 hours, where it gets washed out from the kidney and the spleen rather fast. We then further applied some spectral unmixing methods that allows us to unmix the spectra of the capped dye from the uncapped dye, as well as from the intrinsic background signal. And this resulted in the colored images you can see here in the photoacoustic imaging. So, okay, now we did know that the dye actually enriches in our tumor, but we did not know if it's inside our tumor cells or in the extracellular space. So to investigate this, we did some histology and microscopy with the tumors. And as you can see here, the dye mostly accumulates in the non-necrotic areas. And if you could look further in here, you can see that the dye actually is in the cytosol of our cancer cells. We then wanted to further counterprove that when the dye is inside the cancer cell, it actually gets converted from the capped dye into the uncapped dye. And therefore, we have set up an LC-MS method that enabled us to measure the relative amount of the capped to the uncapped dye into tumor as well as in the liver. And as you can see here, so we only have injected the capped version of the dye, but with the LC-MS, we could always detect also the uncapped dye in the tumor, meaning that plotting it like here, around 35 to 40% got converted from the capped version into the uncapped version inside our tumors. When doing the same with the liver tissue, we could see that less than 5% got converted. That means that our dye was really specific to pathological hydrogen peroxide concentrations and did not show the same reaction in kind of healthy tissue in our liver. So to summarize the work, we have designed, synthesized, and characterized an hydrogen peroxide activatable contrast agent for photoacoustic imaging, and have evaluated it in vitro, in vivo, as well as ex vivo. In future, we should now explore more structural modifications to get a greater spectral difference between the capped and uncapped dye, and to attach a better targeting structure, as the glucose was a more preliminary cancer targeting structure. If this is all in place, then we should investigate better unmixing approaches and fluence corrections. And if this also succeeds, we can then go ahead and combine this contrast agent in a therapy monitoring study with our intrinsic signals of oxy and deoxyhemoglobin to get a readout of hypoxic stress and oxidative stress simultaneously throughout the whole cancer development process. And with this, I want to thank all the groups which helped me with this project, especially also Ithera Medical for their great support. And of course, I would like to thank you for your attention.
Thank you very much, Judith. That was a very interesting one that has generated quite a few questions, which we'll address at the end. Sorry. And now what I'd like to do is hand over to Alexa. So I'll just share the screen now to Alexa, where she can introduce her work at the University of Münster.
Can you see my screen?
Can indeed, Alexa.
Perfect. Okay, as Tim already said in his nice introduction, I'm still doing my PhD about optoacoustic tomography of inflammatory activity in early stages of arthritis using an S109 specific tracer. First, I would like to give you a short introduction about rheumatoid arthritis. It's a chronic progressive inflammatory autoimmune disease, and it has articular and systemic effects. And the symptoms are joint pain, stiffness, swelling, and a decreased movement. And first, we have a metabolic changes in rheumatoid arthritis, where we have the massive infiltration of mononuclear and polymorphonuclear cells into the joint, which is called synovitis. And in later stages, we have also morphological changes, where we have destruction of cartilage and bone loss. And these changes are irreversible. In the moment, the standard imaging methods employed for the diagnostics in arthritis are X-ray, MRI, and ultrasound. And here you can just see the irreversible later changes of rheumatoid arthritis. To see already the metabolic and molecular changes in the early stages of arthritis to prevent the irreversible changes later, we need molecular imaging techniques. And molecular imaging techniques we could use is PET and SPECT, but here we have a high radiation exposure and also high costs, and optical imaging, but here we have not a really deep penetration depth, and therefore we can't image inside the joint, and optoacoustic, which could be a tool and which I try to test in my thesis. Yeah, we used S1N8 and S1N9 as a biomarker for the synovial inflammation in arthritis, and S1N9 and A8 and A9 are expressed and released by activated phagocytes, and they are early amplifiers of the inflammation by the activation of TLF4. In our lab, we have a small non-peptidic specific ligand which binds to S100N9, which is coupled to iodide 800CV. And then you can see the extracellular S100N9. Yeah, as a mouse model, I used antigen-induced arthritis. And first, you have to do an immunization of the mice, where you inject MBSA and CFA and Embodada pertussis. And then you do an intra-articular injection of cationic MBSR into the knee joint. And there, the positive charged MBSA accumulates to the negative charged connective tissue. And then via an indirect activation of EI1 beta, you get rheumatoid arthritis. One advantage of this animal model is that you have the inflammation in one defined knee joint, and the other one serves as the control. First, we did unspecific FDG PET-CT imaging of the mice to see if they have if they got an inflammation and the animal model works. The uptake of FDG by tissue is a marker for the tissue uptake of glucose, which correlates with the tissue metabolism, and this is increased in inflammation and cancer, for example. And here in the upper row, you can see in the inflamed knees that there is a high concentration of FDG and high signal in the synovia, where we can also see in B, the red part in the schematic view is the synovia, and we can see in the FDG can nicely that there's an inflammation, which we can't see in the lower row, the control knee. What we also see in the control knee is the signal in the bone marrow, but this is there's always an activation, which is also in both knees a little bit increased due to the immunization in the animal model. In addition, we did a quantification of the FDG scans, and as you can see, there's a significant difference between the control knee and the inflamed knee. But what you can also see is that some control knees has a higher signal than the inflamed knees, which is due to the spill-in of the bone marrow signal. Next step was optical imaging to first see if the tracer binds to S109 in our animal model. And in the in vivo images, you can nicely see an accumulation of the tracer. In the upper row, we have the inflamed knee and the control knee in the lower row. In the in vivo images, it's hard to see where exactly the inflammation is located. But if you see the ex vivo images, we can nicely see that the whole synovia is inflamed. The quantification confirms these results, and we can see a significant increase from the control knee to the inflamed IR knee. Next, I did optoacoustic imaging of the same animals. We can see a slice here from the height of the knees. There's the control knee and the inflamed knee also in the increased view, and we have a nice signal in the knee joint, in the inflamed knee, and no or almost no signal in the control knee. And also here, the quantification confirms that we have a significant increase in the inflamed knee compared to the control knee. There's also a correlation of the optical and the optoacoustic imaging. Of course, this correlation is not perfect because we compare 2D and the 3D method, but clearly we can see that the control knee has a lower signal and a higher signal in both modalities in the inflamed arthritic knee. To prove a specific spacer binding, we did a blocking study where we injected unlabeled substance 1 hour before tracer injection. And as the image looked before, we have a nice signal here in the inflamed knee, no signal in the controlled knee, and in the blocked animals, we don't see the signal in the inflamed knees anymore. The quantification confirms this as well. We have an increase of the knees which are not blocked and a significant difference which is on the level of the control knees and the blocked areas. Next, we did therapy monitoring in psoriatic arthritis, which is a different animal model, which is very heterogeneous as well. There, the arthritis can develop everywhere. We use black 6 mice, which are, have a genetic background that they develop, that they express HTNF if you apply doxycycline. And as a side effect, the mice were white. Even if it's a black 6 background, you get white mice, which is very nice for optoacoustic imaging. And due to the overexpression of HTNF, the mice develop psoriatic arthritis. The doxycycline, which induces the overexpression, was applied over 28 days. And at day 14 and day 28, we were imaging the mice. And then we started also a treatment from etanercept and PBS in the control knee group as at day 14. First, we were able to see nicely the inflammation in the paws, in FRI as well as in MSAT. You can also see like small spots like in the finger, in the FRI as well as the MSAT. In day 14, at day 14, you can still see like a high signal, which was decreased at day 28, and where the spots were gone. The quantification showed the same. In the PBS treated group, we had an increase of the signal from day 14 to day 28, where we had a decrease of the signal in the etanercept treated group. And the relative change from day 14 to 28 in PBS was the signal increased around 50%, and in the etanercept treated group, we had a decrease around 10%. To summarize everything, we were able to detect SRI9 in the antigen-induced arthritis using our tracer IRDI 800 CV test. We were able to do optoacoustic imaging of the inflammation. We could also quantify the inflammation, and we were able to see a correlation between MSAT and IVA signal. And the blocking of S109 signal proved the specific binding of our tracer. And furthermore, we could also do monitoring of the therapy and over time. Yeah, I would like to thank my whole work group and everyone who helped me during this time.
Brilliant. Thank you very much for that summary of your work there, Alexa. Again, this is generating questions, but while we're processing those, I'll hand over to our final speaker of the day, which is Cheryl Roberts over in New York. So good morning to you, Cheryl, and here is the screen.
Good morning, Tim. Thank you. All right. Yeah, so hello, everyone, wherever you may be tuning in. It's great to be here. And I thank Isara and Tim for hosting this webinar session for us. Today, I'm going to very briefly talk, or rather give a crash course about imaging the glucagon-like peptide 1 receptor with optoacoustics. So first and foremost, I have no conflict of interest to disclose. And I thought I would start with something, I thought it would be great to share with you one of the things we do during our lab meetings recently, since we are all at home, to keep it a bit more interesting. And that's, we share a few pictures of what life was like before during COVID-19. And I thought I would do the same here to start. This is our lab before COVID-19. And now, we are solving a lot of scientific problems, one Zoom calls at a time, or in this case, in our case, one webinar at a time to communicate our work. So thanks again for tuning in to this webinar. During our panel, we have heard some fantastic examples from Dr. Weber and Dr. Hasenbach of how optoacoustics can be used as a tool to study cancer and inflammation. And this can be expanded to many scientific areas that Tim had mentioned also, like diabetes, which is what I'm going to talk today. And we want to do this because the burden of diabetes is great and is associated with increased risk of other health issues, including cancer. And it's becoming a global health problem, and not just in developed countries here. There are millions affected with diabetes today, as you can see clearly here in dark orange on this graph. And those numbers are expected to substantially increase in the next few days, as you can see in the light orange here. And you can see how globally deficit it is in all countries across 155 nations, including developing countries as well. So because diabetes is increasing rapidly, it is expected to drive the demand for molecular imaging in order to facilitate diagnosis and monitoring of the disease in the future. So the current and main effective method for the detection of diabetes that is still used today is glucose sensing in the blood and plasma samples, such as the AC1 test, in combination with family history of diabetes. So on top of that, non-invasive glucose sensing has been tested in optoacoustics as well. A few examples, if not all, I've listed here have emerged in the last 4 years, in case you want to dig out into recent literatures. So this is really like very few literature studies done on this. In general, though, there are drawbacks to imaging glucose. We think of glucose as an indirect way to measure the amount of glucose in the blood. In fact, though, there are other ways to probe diabetes, and one of them is to image beta cells. And why? Because these cells are responsible for the sole responsible for the regulation of insulin and ultimately sugar in our body. As well as that, though, there are also drawbacks to imaging the beta cells, which have really limited and long challenged the imaging of false pancreatic islets in animal models. And these drawbacks I've listed here. The tracers have to be specific to the beta cells. Second, there are very few of these beta cells in the pancreas. And the third is that the beta cells are located deep in between organs of clearance pathways. So to address some of these issues, this is really where we think optoacoustic technology can help and advance some of the challenges that we are currently facing. So we think of this because optoacoustics, which combines line and sound, we think is the ideal choice because we want to go beyond the limited depth imposed by other optical techniques just now, as you can see here in this graph. And optoacoustic can span in here in the middle across different resolutions. It can span across reasonable resolutions in combination with high spatial resolution. So I hope you can appreciate that here in this graph. And in the last few years, we are actually familiar in our lab imaging the glucagon-1 peptide 1 receptor in our lab, headed by my mentor, Thomas Reiner. We have accumulated several experiences now working and imaging the probe for the detection of beta cells. These studies span from fluorescence here, as you can see, PET, we have evolved through around PET and also like PET fluorescent multimodal imaging, such as this bioconjugate paper here from my colleagues. And in more recent years, I have also developed new PET probes for indium-111. And more recently, we have moved on to optoacoustic as a tool, because as I have mentioned before, it could be useful for the challenges that I mentioned. So today I'm going to talk specifically exactly on those topics. And our favorite target for beta cell imaging is the glucagon-like peptide 1 receptor, as I have mentioned. And this is because these receptors are present and abundant in beta cells, on the surface of beta cells. There are a wealth of information available on targeting this particular receptor. And extended 4 in particular, shown here in blue, is a peptide also known as excitinide in the clinics, and it's an agonist, and is the only drug approved for type 2 diabetes treatment currently. What I'm showing you here is in orange is the extracellular domain of the glucagon-like peptide 1 receptor and how this binds to extendin 4. And the favorable pharmacological properties of the extendin qualifies these as our lead tracer for beta cell imaging, as I've shown as an example. And we have been working, our lab has been working on this for a few years now. At the 12th position of the extendin 4, shown here in orange at the bottom, we created a new peptide which we can, which we have modified, which has an alkyne modification. And to go into more specifically, this amino acid sequence is modified on the 12th position, as I've shown here. The scheme is showing you the last synthetic step on the left-hand side, which is the bioorthogonal copper catalyzed quick reaction with cyanine 7 as our optoacoustic tracer of choice. Here are some of the photophysical properties, but basically what we know is that our agent binds specifically shown here in red, and it gets internalized. And the green is showing you the nuclear staining of Min6 cells with NucSpot 488, and the nuclear staining is suitable for we use this nuclear staining suitable for near infrared molecules. The cell line we have used is Min6 interleunomus cell lines, because these are cells. These are beta cells that are expressing normal amounts of glucagon-1 peptide receptor as in humans. And what we have also done is that we have determined its IC50 values measured here in a competitive assay, and we have shown that there is no significant reduction of the glucagon-1 peptide 1 receptor target binding, and these numbers are comparable to the extendin 4, which is around 3 nanomolar. And I've shown you here what we have also done is a blocking study where we have blocked with five-fold excess of the extendin 4, sorry, and you can see that there are no fluorescence shown in this graph. In addition, we have also we further show that the accumulation of our extendin 4 analog tracer is the glucagon-1 peptide receptor mediated via this experiment. We have this is a representative epifluorescent images in mice that we injected either with saline, with blocking, and a contrast agent in the right-hand side showing the fluorescent, and on the right-hand side shows you the fluorescent quantification. So we show here that we can block these signals. The region of interest is the quantification, as I mentioned, and here the size does not grow because it's in chillinoma, and the hotspots that you see around the circles are the organs of clearance pathways, such as kidney, liver, and spleen, and the bottom hotspots are urine secretion, perhaps. What we have also done to verify the true optoacoustic nature of a signal from the agent, we took a look at the kidneys, an organ of clearance pathway where the agent typically accumulates, and here we have extracted the spectra obtained from the ROI around the kidney, and we have shown that its optoacoustic spectra is very similar to what we have done with our phantoms. So talking about phantoms, we have created this soft tissue mimicking phantoms for our MTOT, and we tested different concentrations of our dye. These are phantoms based on work by Cook in this reference here. And here where we can see on the right-hand side is an example of MTOT acquisition carried from 680 to 900 nanometers at varying concentrations of our extendin 4 tracer. The spectra shown here is an example of an image reconstruction after a multispectral in the middle, and then the merge between the background and the composite between our background and our tracer. So we can see that we can multispectrally and mix this really well. In our in vivo setup, our animal model is a hairless female nude mice between 6 to 8 weeks old. We injected 1 million MinSig cells into the flank, and we imaged this mice before and after a 30-minute injection of our tracer. So what you're about to see is a video, and I hope you can appreciate the difference between the pre-imaging and thought and post-imaging and thought. And just before I start the video, I want to mention that we choose this model to focus on reproducibility because in order to reduce complexity that comes with imaging the pancreas, as I have mentioned in the beginning, and to do this, we want to minimize the physiological challenges that exist already imaging the mouse pancreatitis, and because in the mouse the structure of the pancreas in the mice is actually quite different from the humans, and they are rather flat and then dendritic-like, whereas in humans it's a solid mass. Still, in our studies we have demonstrated also uptake in the pancreas. So here what I'm going to show you is the 2D projections of the mice around the flank where we originally injected the cells, and hopefully you can appreciate these signal changes. So you can see here this is mice with no injection, and here mice after the same mice after 30 minutes post injection. And here I want to show you these images in stationary, and so you can appreciate how much signal difference we get. And this is also shown in our quantification on the right hand side. So I went a bit too fast on that, but what we basically want to do is because what we want to carry on doing that while we have shown that we can actually image these beta cells in vivo, we want to also continue with some future perspectives using the rasterscan optoacoustic mesoscopy, which have gained a lot of attention in recent years. And what it does is that it typically excites in a wavelength like the 532. And what I'm showing you here is the same mice, MIN6, and imaged between 11 and 100 megahertz. And what you can do with the RSAM is split these frequencies. So because the signal frequencies correspond to the absorber size, we can do a variety of things as, for example, splitting these band frequencies in various ranges, such as high and low, to be able to differentiate between large and small vessels, as you can see here between the red and the green, and to resolve and the smaller, the higher frequencies can resolve smaller structures. And there are several challenges in using the RSAM for our work. One is that we need to be away from the blood, because at 532 we can only image blood, and although these studies are great for that, for example, like tracking vasculature formation around these MIN6, we want to be able to define our tracer signal away from blood. And the first one is the advantages of doing tracking formation over time is that in recent years there's some improvements in grafting beta cells into patients, where you would inject sub-Q beta cells to help improve insulin regulation in patients. And that's been an ongoing work in recent years. And this would be great to basically track vasculature, because the vasculature is so important for these cells to be to survive. And typically what happens in these methods right now is that it forms necrosis and the cells die. So therefore, the treatment of diabetes cannot continue. So tracking this vasculature over time is an important factor. But the second one is we also want to define a signal tracer away from blood. And those are the challenges that I'm currently investigating, as well as future work that could include with these studies are the 3D optoacoustic tomographic systems, which could work in favor with the near infrared absorbers. And the handheld systems are known to provide quantitative assessments as well with greater sensitivity than 2D systems, which could be up for discussions in this seminar. And with that, I just want to conclude my work. So we have provided the first successful chemical design and synthesis of the glucagon-1 peptide 1 receptor targeting probe, which allowed us to image optoacoustic imaging of beta cells in vivo. And this could be a platform for us to improve further our imaging technique with optoacoustic, and we envision that similar optoacoustic probes could be designed based on what we have started with. And for the reasons that Dr. Weber have mentioned, we have used size 7 in our systems because they work really well with optoacoustic systems right now. And with that, I would like to conclude my talk, and I'd like to acknowledge my PI, Thomas Reiner, and the entire lab. It's a great lab to work with, and this is the Department of Radiology here. And I would like to end with thank you for your attention, and I'll be happy to take your questions.
Thank you very much, Cheryl, and thank you very much to all of our speakers today. So that was Judith Weaver, Alexa Harsenbach, and Cheryl Roberts. We do have a fair few questions come in, so we'll answer those for the next 10 or so minutes. And if you do have any other questions, please feel free to type them in and we'll try to get to them. So I'll start with an easy one for you all. So what I'll do is I'll ask a question, and if you have a particular opinion, please feel free to answer it. So the first question is, what would make the ideal optoacoustic imaging agent, and can this work on optical as well?
Shall I go first, or
go for it, Cheryl. Jump in.
Yeah, so that's a good question. Since I came to MSK, I've been trying to figure out whether dark quenchers are actually better than the fluorescent ones. And the main reason why we keep going back to fluorescence is that we can validate our work through optical techniques as well. And a lot of reviewers like to see cell studies where we can actually prove what we are looking at and what we are targeting. And unfortunately, with dark quenchers, we can't do that, and we are limited to optoacoustic to validate our work, and it requires a lot of attention to detail. So there are some studies that I've published that show that dark quenchers are better, but these are few examples which we cannot conclude really so far.
So I guess that quenchers, while giving a very, very strong optoacoustic signal, they give a minimal optical signal. So it does make validation difficult. So probably the ideal chromophore or fluorophore would be one that still has some emission properties for optical as well.
Yeah, exactly. And that's why we went with something like a cyanine 7, where we know it kind of works, and we can also validate this through IVIS imaging.
Okay. A general question, actually, I think this is probably more targeted toward Judith, but this is to do with the absorption spectra from fluorophores and optoacoustics. Optoacoustic spectra seem different than fluorophore excitation spectra. Is there a reason for this? And just a very specific question to your work, is the absorption spectra change from capped to the uncapped probe look very similar to the change in dye absorption during aggregation and disaggregation? Could you comment on this?
Yes. So for the first part, I mean, there's always the way how the signal is produced is via the conversion right into the sound wave. So you will always, at least from my experience, you always see this broadening a little bit of the spectra, and which is also caused by the environment and everything. So for my dye, yes, I had the same doubts that it looked at the beginning to me as an aggregation spectrum, but I proved it by doing dilution steps and also further investigating why this kind of spectra is kind of caused with computational studies. And actually, the computational studies also confirmed this kind of double-shaped peak and the way how it changed by kind of bending my dye backbone when something on the top gets released or not. So yeah, my first concern was also with aggregation in aqueous solutions, but this wasn't the case. At least, so you should also see then a concentration effect if you would see this, and fluorescence should go down at once when they aggregate. So yeah, for my dye, I would say, I hope, I would say it's not aggregation based, this double-shaped peak.
Okay. So I'll turn this into a general question for everybody. So this was we've had the question for inflammation and for diabetes. So specifically for diabetes, we've seen that you can do BAT imaging using MSOT, which I assume is brown adipose tissue, using HbHbO2. And a similar question for inflammation is that you can use HbO2 to measure inflammation as well. So using a technology like MSOT, can you validate your optical, so your contrast agent signal, with endogenous signals like change in HbHbO2? So specifically in inflammation or in diabetes?
Yeah, so I am aware of the brown adipose tissue imaging. Yeah, I guess we could. And the most important thing with that is that the spectral mixing has to be really specific to that tissue that we are looking at. And that is good in one way. But the thing with brown adipose tissue, I haven't worked on that, I have to say, but I can imagine that you know, you would have to have, you know, what's great about endogenous contrast imaging is that mainly we are imaging blood, and there's plenty of blood to image. And with brown adipose tissue, I'm not sure how much of that we can really see very small changes, like for example the beta cells that are decreasing over time for diabetes patients, for example. So that's what happens over time, right? Diabetes, people are having problems regulating insulin because the beta cells are dying over time, and they're very slow, and there's really small amount of changes. With brown adipose tissue, I'm not sure how. I can imagine that it works well as well for optoacoustic imaging. Yeah.
Okay. And if Alexa's still on, what would you consider for imaging inflammation?
We saw also an increased signal in the Hb and HbO2 mixing. And to prove that we don't just image the perfusion and the increased blood signal, we did the proof of the specific binding with the blocking. But I didn't really look so much into it, but there was also definitely an increase in the Hb and HbO2.
Okay. And then while I've got you on, Alexa, can you evaluate the etanercept in the short term?
Sorry?
Just a question. Can you evaluate the etanercept in the short term?
Yeah. It's supposed to have a very, so there's another study where they already treated the animal model with etanercept, and also looked at the skin disease, which is also from psoriatic arthritis. You also have the psoriatic skin defects, and therefore we already knew that you have an effect in 2 weeks. And now my data from just the arthritic side showed the same. So there was definitely an improvement after 2 weeks.
Okay. And then we'll ask this as a general question, because this has been commented for all the speakers, is in terms of quantification, are you comfortable using MSOT as a quantitative method to, for example, determine inflammation or enzymic activity or number of cells and so on? And is this an advantage over, say, optical?
I can comment on that, and I have a lot of experience. So I think of optoacoustic as a semi-quantitative measurement. So I always go with this, like what I've shown here, before imaging and post imaging. And this is because when you do multispectral and mixing, there's always a score, which is a signal, no matter what you do. So you can have false positives, for example, which is at the moment quite prevalent in multispectral and mixing. So like I would use it as a semi-quantitative. It's not like fluorescence where you either have a signal or not, because you also have endogenous signals that are absorbing, as well as your contrast agents that you are looking at.
Okay, and maybe Alexa, are you happy with your results in terms of quantification?
I would say the quantification in the MDOT was as good as in the optical imaging. I also had like sometimes artifact which maybe had an influence, but in optical imaging I had the problem that sometimes there was the inflammation more on the left side of the knee and sometimes on the right side. So depending on how you did the optical imaging, there was also a different signal. And with MDOT I had like the whole knee imaged. So this was an improvement. And for example, with the quantification with FDG, I also had my problems with bone marrow. So I would say it performed as good as the other methods.
So regarding actually imaging, I mean, I suppose this is for Judith, you're looking at potentially a very complex environment. So for peroxide sensing or detection of oxidative stress, how quantitative or how happy are you with your kind of data?
That's a difficult question because, as I pointed out in my last slide, that my dye is not optimal. So it needs improvement in structural modification to get a better difference in the spectral shape between the cage dye and the uncaged dye. So I guess I mean, the results show the same trend as my LCMS study. And so it still shows that it has the same direction. But as I pointed out, this needs to be improved with the structure. And then I need to get some better unmixing approaches and fluence correction. And if this all will be successful, then I think it has great potential. So I think, yes, with my dye, I would be a bit more skeptical at the moment, but mostly due to the dye's properties so far.
Okay, so more of a preference question now is, do you prefer imaging in optical and then MSOT, or do you prefer MSOT and then optical?
Definitely optical and then MSOT, because you have to really clean the mouse really good, because otherwise you have like some reflections from the ultrasound.
I always did before and after, and it worked equally nice.
So you imaged.
Yeah, I started with fluorescent like iris, then I imaged the mouse, and then I imaged again with the iris.
Yeah, this is something I do very similar to this. Yeah, I do the same as well. I would image with the iris, track it, and then if everything goes well, I would image it with the MSOT, because also the optical imaging techniques are much faster to probe what you're looking at, and then you can go ahead with the optoacoustic imaging setup. And the danger is that, you know, sometimes with optoacoustics, since we are, especially with the MSOT, we are working with water, and yeah, there's some challenges there.
Okay, so possibly that comes down that optical, people have asked this question, so optical is a good screening tool, but then optoacoustics for kind of depth of information or quantification. Yes.
Like definitely for me, because I could not get the same readout with fluorescence as I could get with the photoacoustics due to the spectral information.
Okay.
Yeah.
So general question, yes, mice obviously need to be shaved for fluorescent imaging, and they also need to be shaved for optoacoustic imaging. That is something that needs to be done. And I'll kind of try and wrap up. I'll just pick a nice question here. Still got quite a few to answer, but as it's now on the hour, we should stop. And that is, Alexa, you show very high resolution images of the knee. What is the resolution you achieved with MSOT, and was the bone an issue?
The bone was not an issue. And even if there would have been reflections from the bone, it would have been like a reflection of the true signal, which is left to the bone, next to the bone. And the resolution was really good. I didn't show this data, but also, like when I imaged one day after the injection, I had I made a small cut in the skin, and there's also like a small inflammation where you also get S100M9 signal, and I could even distinguish together with the ultrasound data from the MSOT between the small cut in the skin and the inflammation in the synovia.
Wow. So, I mean, at least we state that the MSOT resolution is 150 micron and the RSOM resolution is 10 micron. And I think your images do show that. So some more questions we will follow up with offline, but I want again to say thank you very much to all of the speakers for taking the time to present their work today. I thought it was really informative and of very much interest to the molecular imaging community. Thank you all for attending and for bearing with us for the last hour. As mentioned, we will send a recording of this video out in the next couple of days, and we'll always welcome you to our next webinars in the not too distant future. So thank you very much and have a very good rest of your day.
Thank you.
Thank you. Bye.
Thank you. Bye.